Sometimes genetics does not follow the normal rules that Mendel discovered. One of those ways is with multiple alleles. Normally, an allele is either of two alternating forms of a gene for a trait. So, for example, you might have the allele for widow's peak, or you might have the allele that says no widow's peak. So you only have those two options. Sometimes there can be more than just two options. Take, for example, blood types. With blood types, there's an A option, there's a B option, and then there is an O option. So now we've got three different possible phenotypes. Phenotypes are just the physical expression of the genes. All right, genotypes are the combination of the alleles. All right, so what do you have? Two dominant letters. Do you have an A and a little a? That's the genotype. The phenotype is physically, how does that look? So with blood type, there can be more than just two options. It's not, do you have the A protein, and then you might have the not A option. Well, now there's this other B option available. You could have the A, or you could have the B, or you could have the O option. So if you look at my little table here, you can see the relationship between genotypes and phenotypes for A, B, and O. It's a way of typing blood. So for example, if you have two capital letter A's, that's going to make you have the type A blood. If you have two B's, that's going to make you have the type B blood. If you have the two little I's or the two O's, that's going to make you have type O blood. Let's see what that's actually referring to, the phenotype. So here's another little chart. This chart's just showing what that phenotype looks like. So type A has the triangle shape on the surface of the red blood cell. Type B has this circle shape on the surface of the red blood cell. All right, what does that mean? Those are called antigens. So type A blood has type A antigens. Type B blood has type B antigens. I kind of think of it like an identity marker. All right, so you might carry around something that looks like this, an ID. That ID is kind of like your marker. All right, so that's a protein that is on the surface of the blood cell. So if you have the protein, you have the phenotype. All right, the only way to get that protein is to have the right combination of genes, the genotype. So if you have type A blood, you have the type A antigen. If you have type B blood, you have the type B antigen. If you have type O blood, then you don't have either antigen. All right? Now, what makes this interesting is whenever you're donating blood. All right, so to donate blood, a type A person can give it to another type A person. But a type A person can't give their blood to a type B person because the antigens, the markers on the red blood cell, are recognized by antibodies, proteins as part of the immune system, kind of like security guards, that those antigens are foreign. So for example, if I have an ID and my ID is for a school, but I go to a neighboring school district and show my ID, they'll recognize that that ID does not belong to this school. Likewise, if someone came from another school with their ID and tried to show it, our school would say, you don't belong here. That's like having the type A antigens being recognized by the security guards, the anti-A antibodies. All right, type B blood has anti-A antibodies, security guards. Type A blood has anti-B security guards, antibodies. Type O does not have any identification. So the body can't recognize it as foreign. So a person with type O blood can give their blood to type A, and they can also give their blood to type B. That's why they're called the universal donor, because they can give their blood. They don't have any markers on the outside. So let's say we were doing some Punnett squares with blood types, what would those look like? So let's do a couple examples for multiple alleles. So let's say that we have a person who is type A blood, but they're heterozygous, so they're going to be AO. And we're going to cross them with a person who has type B blood. And we want to know what are all the possible genotypes for their offspring. So we'll put AO across the top, we'll put BB along the side, and then we just simply combine them. So I've got an A, B here, I've got a B and an O here, an A, B here, and a B and an O here. So in this case, there's 50% chance that they're going to be type AB blood and 50% chance that they're going to be type B blood, or because O is recessive. All right, let's give another problem. Let's say we have here a person who is B and O and a person who is O and O. In this case, we're going to put the Punnett square together, and we'll put the BO across the top and the OO along the side, and then we just match them up. So we've got a B and an O here, and we have a B and an O here, and we've got OO and then O here. So in this case, there's 50% chance that they're going to be type B blood, and there's 50% chance that they're going to be type O blood. Now you might have recognized here that there's 50% chance that they're type AB blood, and there's 50% chance that they are type B blood. You might be wondering, what's this AB? Well, AB is a different type of trait. It's a codominant trait. All right, let's just do one more. Let's say the person is type A blood, but it's AO, and the person is type B blood, but it's BO. In that case, we cross here, we'll have AO across the top, BO along the bottom. Type AB is possible, type B is possible, type A is possible, and type O is possible. So all the blood types are possible. Whenever you're working on problems like this, you have to be aware that you can work backwards, and that sometimes a person is type A blood, but they're carrying the O allele. All right, so let's go with this working backwards idea. I know that a person has type O blood, all right? Is it possible for the parents to not have type O blood? Well, I can put an O here, and I can put an O here. But if I put an A here and a B here, the parents do not have type O blood, but it is possible for one of the children to have type O blood. So we can work backwards, and we need to be aware that because O is recessive, a parent doesn't have to have type O blood. But if both parents have type O blood, can the child have type A blood? So you figure out that problem and see what the answer would be. Another case where genetics does not always follow Mendel's rules are sex-linked genes. That is, they're genes that are linked to the sex chromosomes. These are generally carried on the X chromosome, so that a female who has XX can have two alleles, but a male who is XY only gets one, because it's carried on the X chromosome, but not on the Y chromosome. So it's linked to sex. All right, in this case, we have the pink X is representing the trait. All right, so the mother is a carrier for the trait. She does not have the trait, but she's a carrier for it. The father is normal, but he's not carrying the trait. And you can see in my Punnett square here, we've got the X's come together from the sperm and the egg, and then the Y from the other sperm and the X from the egg, and neither of those two have the trait. But if the sperm fertilizes the other egg, and it happens to be carrying an X chromosome, we've got two alleles there. So the trait is just going to be carried, but it's not going to show up. In the case of the son, because the trait is on the X, and then there's a Y, like a recessive trait like this is going to show up because there's no dominant trait to cancel it out. So these are more common in males. So if you're looking at a pedigree, for example, or you're looking at something else, you're going to see traits that are sex-linked show up more frequently in males than you'll see them in females. So let's work on some problems that have to do with sex-linked traits like color blindness or hemophilia. Now let's look at the case of sex-linked traits. Sex-linked traits are things like color blindness, for example. So the trait is carried only on the X, not on the Y. So whenever I put together a Punnett square, I'm going to go ahead and put the XX across the top, XY along the side, and then I'll put the trait on it. So let's say that the mother is a carrier. So we're going to put capital B, lowercase b. Color blindness is recessive. So the lowercase b is going to stand for colorblind. Let's say the dad is normal, so he can see normally. In this box, I'm going to put two X's. In this box, I'm going to put two X's. Down here, I'll put an X and a Y. Here, I put an X and Y. So there's 50% chance that you're going to have a female, 50% chance male. But will they be colorblind? So this one is going to have two capital B's, so that daughter will be fine. This daughter will be a carrier. This son will be normal, but this son will be colorblind. So there's a 50% chance that you're going to have a boy. And if you have a boy, there's a chance that he's going to be colorblind. So with these two parents, there's a 25% chance that you're going to have a colorblind boy. Now, let's look at a case where the father is colorblind and the mother can see normally. She's not a carrier. All right? In this case, I'm going to put the girls across the top, boys here, and now we're going to see what combination of genes can we have. So in this case, the daughter is going to be a carrier. The son will see normally. This daughter will be a carrier, and the son will see normally. So let's do a problem where we're working backwards. All right, we're going to put our XX across the top, XY here. We'll go ahead and fill in these X's for girls, and then this XY for boys. When you're thinking of these traits, think of the X's like chromosomes that look like this. The trait is here. So the Y looks like this, and the Y does not have that part of the chromosome. That's why they're called sex-linked traits, because the trait is on that part of the chromosome, but that part of the chromosome is absent for the Y chromosome. In this scenario that we're working on, we want to have a colorblind girl. What does it take to have a colorblind girl, to have BB? If this is the case, it automatically means that the man must be colorblind. It also means that the woman has to be at least a carrier. Now, if she's colorblind, that would guarantee that all the children will be colorblind. But if she's a carrier, it will guarantee that half of the girls could be colorblind and that half of the boys could be colorblind. So the probability is 50-50, but only if the mother is a carrier and the father is actually colorblind. So hopefully those problems help you figure out a little bit more about colorblindness. So here's just a generic colorblind test. Most colorblindness doesn't mean you can't see color at all. It just means you have a hard time distinguishing between two colors. So you can take a look at these images and just see if you can tell what the numbers are inside the images. As a little, it's not diagnostic, but just to kind of see an idea of what colorblindness looks like. So I hope that kind of helps you get an idea of multiple alleles and sex-linked traits.